Okay, welcome everyone to the third in our EV flow cytometry webinar series. Today our speaker is Edwin Vanderpoel from the Amsterdam Medical Center. First, let me start by reminding you that there will be a discussion and question and answer happening on the Slack app that this group is hosting, so check that out to follow the discussion during and after the webinar. So Edwin Vanderpoel, as I mentioned, is at the Medical Center. Known Edwin since the earliest days of these EV flow cytometry meetings, so probably a decade, when he was a graduate student, I believe, with Nolan. And his work was notable for taking a careful quantitative analytical approach to measuring EVs by flow cytometry and other methods. He's very prolific, a lot of very nice papers comparing different methods used to characterize vesicles, including flow cytometry. One of the areas where he's really made some contributions is in the area of light scatter of EVs. Light scatter is a often misused and abused parameter, mostly owing to the fact that it's more complicated than people initially realize. But Edwin, along with my co-host here, Josh, have done really nice work about showing how one can effectively use and interpret light scatter data, EVs. And so today, Edwin's going to tell us about some of his recent work in that area. Edwin.
Thank you very much, John, for the nice introduction. So I will start with convincing you that if you detect extracellular vesicles, that size does matter. So if you run your daily beat, in fact, this is not a calibration. So I hope to give you some light scattering background to make you understand what is actually a calibration and how you can calibrate one light scatter detector, and even use the information from two scatter detectors with a relatively new technology, FLOSR, to derive both the size and the refractive index. And then I will finish off with discussing some applications and limitations thereof. So, detection of EVs, size does matter. To make that clear, you can see here the size distribution of extracellular vesicles in urine, measured and combined in both electron microscopy and resistive pulse sensing. And as you can see, the concentration of these decreases rapidly with increasing diameter. And this affects the detection because you can imagine if you have two flow cytometers with a different limit of detection, so one at 180 nanometer and the other one to 30 nanometers, these flow cytometers measure already a twofold difference in the concentration, total concentration of EVs. So this means that this affects data comparison, because to compare the EV concentrations, you have to state the size range. So in fact, it's bad practice to state we found 2 times 0 to 10 to the 8 EVs per milliliter, but rather you state we found 2 times 0 times 10 to the 8 EVs per milliliter, ranging from 180 to 600 nanometers in diameter. Now also for data interpretation, size does matter. Here you can see the side scatter versus forward scatter plot measured by a flow cytometer of a plasma sample. And the data are in arbitrary units. So it's very hard to confirm that these are actually EVs or particles in the EV size range. It's common practice in the field to set gates based on polystyrene and silica beads. But in fact, that's bad practice, and I will explain that in the next 10 slides. In the literature, you see very often, unfortunately, that gates set on polystyrene beads are used to select the size range of EVs. They don't calibrate the flow cytometer. So here you can see 4 examples of that, and there are 356 more examples in literature which you can find. In fact, running your daily beats is a very important quality control. You can use it to set up your instrument or check reproducibility. But an actual calibration relates arbitrary units to comparable standardized units. For a flow cytometer and a scatter detector, this can mean that the arbitrary units of scattering are converted to the diameter in EVs, in nanometers, for example. Now, before I will explain how that works, you need to know some basics about light scattering. In fact, 3 things. The first is that with light scattering from particles, the light scattering does not only depend on the diameter of the particle, but also on the refractive index. So I represented that here schematically. You can see there 3 particles of 200 nanometers. The first is an EV with a refractive index of 1.40, silica bead with a refractive index of 1.45, and a polystyrene bead with a refractive index of 1.61. And as you can see, the measured light scattering intensity differs substantially. Secondly, light scattering depends on the hardware of the device with which you measure light scattering, in this case the flow cytometer. So it depends on the illumination wavelength and the polarization thereof. And if you analyze the scattering of a vesicle here in the center of this radial diagram, in this case calculated a 500 nanometer EV with a refractive index 1.4, then you see that to all directions this vesicle scatters a different amount of light. So you can imagine that if you have a different flow cytometer, you measure a different fraction of this light, and you will find a different scatter to diameter relationship. And you see some examples of FSC and SSC detectors of flow cytometers in the field. And third, although this sounds so very complex, you can solve this with Lie theory. So with this theory, you can take into account all the scattering angles and the refractive index of your particle. And this is an example. Polystyrene beads here, those are the symbols, silica beads, the red symbols, and hollow silica beads. And we describe this with Lie theory. As you can see, the theory described it very well. So just in sum, what you need to know about light scattering and flow cytometry, measured light scattering signals depend on the particle size, but also refractive index, and on the flow cytometer, illumination wavelength, polarization, but above all, the collection. Now, let's see what we can do with this theory to facilitate sizing of EVs using one scatter detector. And for that, so I use data from 2 different flow cytometers. On the left, we see the influx. On the right, the LSR. And they measured the same mixture of polystyrene beads. And we're going to pretend we want to set a size gate based on 200 and 400 nanometer polystyrene beads. In the back in green, you can see the histogram of erythrocyte EVs measured by the same flow cytometers. And you can already see that it seems that these bead gates select different fractions of this population of erythrocyte EVs. But without doing a calibration, it's hard to explain why. So let's calibrate. Here on the right, let's first calibrate the BD influx. So here on the right, you can see the calibration. You see the forward scattered light versus diameter of polystyrene beads and Lie theory, taking into account the detection angles and the refractive index of the polystyrene beads, and then we extrapolate to EVs, which results in this green line here. And then we see that 200 and 400 nanometer polystyrene beads gate EVs from 330 to 750 nanometers. Now, we do the same for the BD LSR. Calibrate with polystyrene beads and Lie theory, we extrapolate taking into account the refractive index of EVs, and we see that this BD LSR selects way different EV sizes using 200 and 400 nanometer polystyrene beads, namely 820 to 1830 nanometers. So just to summarize this, you take the polystyrene beads, 200 and 400 nanometer, and within with the same scattering intensity on the BD influx, you measure 330 to 750 nanometer EVs. And on the BD LSR, you would measure 820 to 830 nanometer EVs. So first, polystyrene bead sizes underestimate the actual EV size, and second, different flow cytometers gauge different EV sizes using polystyrene bead gauge. Just to convince you that it's really different for every flow cytometer, you see here 15 different scatter to diameter plots, which we measured in a standardization study. Please don't look at the details, just generally all the plots are different. So polystyrene bead gauge are really tricky. So it's hard to turn your flow cytometer into a particle sizer? No, in fact not. Leonie de Ronde, a PhD student who just, a student who just graduated a few weeks ago, explained this very well in 4 ways in this paper. And 2 of these ways are easy, cheaper than a daily quality control polystyrene beads. And dependent on your flow cytometer, it can even be more accurate and precise than particle sizing technology like NPA. Of course, there is also there is a catch. You may have noticed these tilde symbols here in this table, and this catch is that if you want to determine the diameter with one scatter detector, you have to assume a refractive index of EVs. And to make that clear, I calculated 2 different refractive indices here, so 1.40 plus or minus 0.01, and you see that especially for the larger particles, the refractive index determines largely what size you will measure for a given scatter intensity. So size determination with one scatter detector strongly depends on the assumed refractive index. And now is the question, can we derive this refractive index accurately, and how? There is a way for that, and that's by combining the information of 2 scatter detectors. And to do so, we will exactly use the same approach as calibrating one scatter detector. So here you can see the forward and side scatter versus diameter measured polystyrene and silica beads, in this case by an apogee A50 flow cytometer. And then we will apply mean theory, taking into account the refractive index of the beads, the collection angle of the flow cytometer. As you can see, the theory describes the data well. But it's not enough to do sizing independent of refractive index. So this blue arrow represents this. So if you have a signal of 400 arbitrary units, this may correspond to a 200 nanometer bead of polystyrene, but it may as well correspond to a 320 nanometer bead of silica. Without any foreknowledge of the refractive index, you could not tell the difference. However, if we take the ratio between side scatter and forward scatter, which we name the flow cytometry scatter ratio, or just in short, flow SR, we see that this flow SR versus diameter is independent of the refractive index to about 500 nanometer for the apogee A50. And that's just a little bit larger than the wavelength of the illumination. So this means that we can use this to size the particles, but in this case, independent of the refractive index. That works as follows. You just take the ratio, and if this ratio is, for example, 2.5, then you can see that that corresponds to a particle with a diameter of 200 nanometer. Here you can see another example. Our apogee A50 got an upgrade. Now it's an A60. The side scatter detector became 10 times more sensitive, so the axis changed. But you can see that polystyrene beads, silica beads, and also hollow silica beads and EVs, they all follow this same line. The requirement to use flow SR, nevertheless, is that you have nanoparticle sensitivity on both the forward and the side scatter detector, and that your particles are smaller than about 600 nanometer in diameter, but that's flow cytometer dependent. Mean theory is in fact not necessarily required, because as you can see, you may just measure these beads yourself, apply a Gaussian fit, and use the Gaussian function as a lookup table to relate your flow SR to diameter. And that about sizing with flow SR, but how can we use that to determine the refractive index? For that, we don't only use the flow SR versus diameter relationship, but we go back to one of the calibrated detectors. So to explain that, we take this flow SR of 2.5, corresponding to a particle of 200 nanometer. At this stage here on the left, we don't know whether what is the refractive index of this particle. But if you combine it with the calibrated forward scatter detector here on the right, and in fact, you see here the calculations for only 5 refractive indices, but to do accurate refractive index determination, we make a lookup table of many more refractive indices and smaller steps. And then we relate the measured forward scatter to the calculated refractive index. And in this case, for this 200 nanometer particle, the forward scatter was 90, corresponding then to a refractive index of 1.50. Nevertheless, if we would have measured a forward scatter of 10 arbitrary units, this would have been a particle with a refractive index of 1.40. Another example, imagine the flow SR was 0.5, that corresponds then to a particle of 300 nanometer. Now going back to the calibrated forward scatter to diameter table, then we see that if the forward scatter signal was AP, that corresponds to a particle with a refractive index of 1.37. But if the forward scatter would be around 2,800, then it was a particle with a refractive index of 1.63. Now, the way I explain it now is, of course, pretty slow. So if you automate this and you let the computer perform this lookup, you can do this for all the particles in the measure. So does that work well? So we validated flow SR with a bead mixture. So here you can see a mixture of beads, which we labeled A to F. And here it's on the side scatter versus forward scatter plot. But after applying flow SR, we can relate the side and forward scatter signals to refractive index of diameter. And then you see that population B, C, and D correspond to refractive index typical of polystyrenes, and populations E and F correspond to populations typical of silica beads. Unfortunately, population A was wrongly identified, and that's because it was too close to the noise of the forward scatter detector. So how accurate is this? Here you can see the measured size distributions, and in green the expected sizes. And as you can see, the measured and expected sizes correspond pretty well. The measurement error was smaller than 8%, and the coefficient of variation also smaller than 8%. For the refractive index, we don't know what is the measurement error, simply because we don't have good standards for refractive index. Even of polystyrene B, the refractive index is not exactly known. But the CVs were decent with a value smaller than 2%. In turn, we validated flow SR on polydispersed samples. We took 2 oil emulsions with oil droplets with a refractive index of 1.36 and 1.40, and intralipid, which has a slightly higher refractive index. And as you can see, the refractive index distributions corresponded pretty well to what we expected, although this intralipid was a little bit wrong. Now we have validated flow SR. We can apply it to some samples. So here you can see that we measured the side and forward scatter of particles in the supernatant of an outdated platelet concentrate. It's very hard to draw any conclusions from this, but if we apply flow SR, we can go back to our refractive index diameter plot, and then you get this complicated pattern here on the right. So it took some time for us to understand what happened. This asymptote here, totally on the left, is basically the detection limit of the forward scatter detector. In fact, it means we can detect large particles with a low refractive index and smaller particles, but only if they have a high refractive index. That's why this curve goes up. In fact, we don't expect particles with a refractive index higher than 1.6 to be present in plasma. So this asymptotic behavior is wrong, and we trust flows are only above 200 nanometers in diameter. So looking at this data of particles larger than 200 nanometer, you can see clearly that there are two distinct populations. And our hypothesis was that the lower population corresponded to vesicles, and that the higher refractive index population corresponded to lipoprotein particles. So to confirm, we labeled the vesicles with CD61, the platelet marker, because this was an outdated platelet concentrate. And here on the left, you can see the refractive index diameter plot for only CD61 positive EVs. And then you can see that the majority of particles, 97%, had a refractive index smaller than 1.42. So now then the next question which arises is, what was this upper band here? So our hypothesis was lipoprotein particles, but it was much harder to confirm that, mainly because we couldn't find a good label to specifically label microns. So Leonie, who just graduated, she performed a density gradient, and top down in this case, she collected fraction 1 to fraction 12. She measured the density, and she applied Western blot, electron microscopy, and flow-jar to these fractions. And she showed that by Western blot, that the EV fractions with CD63 were particularly present in fraction 6, at least the peak. And the lipoprotein fractions, which were APO, B100 positive, were mainly in fraction 12, 10, and 12. And also with electron microscopy, she showed that in fraction 6 were vesicles present and 12 predominantly lipoprotein particles. So, and here are the flow-as-R results. So if we focus mainly on fraction 6 and fraction 12, you can see that in fraction 6, which is the vesicle fraction, most particles had a refractive index below 1.42, whereas in fraction 12, most particles had a refractive index higher than 1.42, indicating that those are most likely lipoprotein particles, chylomicrons. So then, what can you do with this? What she studied is, so she labeled the particles in this same sample with different generic membrane dyes, and she checked in which fraction these particles ended up, in the high refractive index fraction, which we attributed to lipoproteins, or in the low refractive index fraction, which we attributed to EVs. And as you can see, we didn't get quite working, but calcium violet resulted in 13% of the chylomicrons being positive. D8 and NAPS, the majority of the particles was at least in the high refractive index band. Lactate seemed to perform better in our hands with 21% of the high refractive index particles stained. And as a control, we also added CD61 APC, which you could only label EVs, and there only 11% of the high refractive index particles was stained. So to some applications, but also limitations of flow-as-R. Applications are size and refractive index determination. You may use that to differentiate EVs from lipoprotein particles. For example, to study marker specificity, we may also use this to increase the assay specificity. Limitations are that flow-as-R requires a sensitive forward and side scatter detector. Most flow cytometers are equipped only with a sensitive side scatter detector. And it works in a limited range only. So for the Apogee A60, that's about 200 to 600 nanometer. This depends on the flow cytometer. And yeah, particles outside this range may cause artifacts. So you need to be sure that the majority of particles is below, for sure, the higher cutoff. As an outlook, yeah, we automated this process. We automated all calibration steps, which we now do, including FlowSR, with a software package which we named Falcon. It consists only of 8 buttons to make it as much as possible. And we apply this in the first clinical study, which is named AFFECT, involving 1,224 FCS files. So obviously you don't want to manually process all those files by FlowSR. I'm not going to talk about this. It's the topic of the next immediate flow cytometry seminar series. So in sum, yeah, if you detect EVs, always keep in mind that the size matters. Ask yourself the question, in which size range am I measuring the EVs? A calibration is not just running your daily beats. No, a calibration is relating arbitrary units to standardized units. And for scatter, a good choice may be the diameter of EVs. And it's doable. You can read the paper of de Rond et al. To see your options. Yeah, for FlowSR, this allows particle size and refractive index discrimination. That sounds great, but please be aware that's only within a limited size range, so about 2 to 600 nanometers. Now, with that, I would like to finish. I would like to acknowledge my colleagues in the Amsterdam University Medical Centers, the EV flow cytometry working group for having very useful discussions over the past years. And Zoltan from the Hungarian Academy of Sciences for providing hollow silica beads. For more information, please check these websites. And I would like to thank you for your attention.
And thank you, Edwin, for a very nice, clear talk. We've got time for questions, and some questions are coming in. If you have questions, add them to the Slack chat. Edwin, can come back and answer those more fully later, but we'll let him have a crack at a few of these right now. I'll start off. So Edwin, is FlowSR applicable for any 2 scattering angles, or is it really work when you have a large and a small scattering?
Yes, that's a good question. I did some calculations for different instruments, and the FlowSR versus diameter relationship differs between instruments of different scattering angles. If the forward scattering angle becomes narrower, then I've seen that the FlowSR to diameter relation becomes steeper, and the maximum range with which you can apply this technique decreases. So in fact, this means you get higher sizing accuracy and a more limited range.
Interesting. Okay, we have another question here. Does antibody labeling of EVs impact on particle refractive index? Can antibody labeling improve the detection of small particles undetectable with no labeling? Questions?
So do the antibody labels affect the particle refractive index? That's a very, very tricky question. That I think the best would be to measure it. But even that doesn't answer the question well, because what I didn't explain is here I'm talking about the effective refractive index of particles, meaning so a polystyrene bead, for example, is a solid particle with 1 refractive index, but the vesicle, in fact, does not have 1 refractive index. A vesicle we measure as a particle having an effective refractive index, but in fact, the phospholipid membrane of the vesicle has a high refractive index, whereas the lumen of the vesicle has a low refractive index. So I can imagine that if you stain the vesicle, that you add proteins to the membrane, and thereby perhaps increasing the refractive index of the membrane, which was already high. So effectively, you may increase the refractive index a bit. Yes, it's just a matter of measuring it.
So it's complicated.
So it's complicated. Yes. Yeah.
So the second question there is, can antibody labeling improve the detection of small particles that you can't detect with no labeling?
Yeah, that's also an interesting question, and this highly depends on the flow cytometer. Generally, I think that fluorescence is easier to detect, but the advantage of fluorescence detection is that you use a spectral filter. So this filters out the illumination of the laser, meaning what's left over is only signals from the labeled EV or background in the medium. Whereas with scatter, besides the scatter from your EV, you will always have some background scattering from the laser and the optics and the flow cytometer. So from that perspective, you would say it's probably easier, or you get a higher signal-to-noise ratio for fluorescence detection. In practice, I think many flow cytometers have this. Ours not. The flow cytometer which we use is about as sensitive for triggering on scatter as triggering on labeled EVs. So it's just a matter of figuring this out for your flow cytometer.
Okay. Okay, a couple practical questions here. One is from me. It said, say I have a flow cytometer, whatever brand, and I want to assess its ability to, my ability to do this. What test should I do, and what will it tell me? Will it tell me, for instance, if I run some beads into this analysis, will it tell me the size limit of detection of my instrument?
Yeah.
And then a related question there, someone asks, how would this work in the image stream, imaging flow cytometer, which has no forward scatter?
So if you're interested in figuring out whether your flow cytometer is applicable to doing flow SR, I would start with doing the experiments which you can see on this slide. So start with well-defined polystyrene beads, measure their diameter, measure their flow SR. So you measure their forward scattering and side scattering intensities. Here I used the area parameter, I took the median, and I simply took the division. What more I didn't do. And I got this result already. So if you get a similar curve like this, you can start with fitting this curve with, for example, a Gauss, and check whether beads of a different refractive index also fall on top of this curve. Generally, practically we have tried this on 3 different flow cytometers, 2 apogee instruments and an influx, and that all worked. And we concluded that for all detectors, it's going to get useful if at least you can detect 100 nanometer polystyrene beads readily from the notch. So if you cannot, the range in which you can apply flow SR becomes very limited, especially because above a certain diameter, these flow SR versus diameter lines deviate from each other, and the technology is not applicable. The Gaussian fit doesn't tell you this, so if you want to know what's the maximum size for your flow cytometer with which you can apply flow SR, you need to do some reviewing. For an image stream, I don't know, does an image stream measure forward scattered light? I think not.
It does not.
No, it does not.
So, okay.
So without forward scattered light, it would become difficult.
Okay, next, I guess another practical question. Ben asks, what are the best options for standards currently? I presume he's talking about light scatter standards. What beads do you use for this?
Which beads do we use?
Does it make a difference?
Now, our most important references, the most important reference beads which we use, and that we always start with, are NIST traceable polystyrene beads. I think they were named Duke beads, and now we buy them at the Thermo Scientific. They are narrow, so they are monodisperse, and they come with a NIST certified diameter. Generally, they are stable for a long time. They come in high concentration, so we can use them quite long. Yeah, the silica beads are of lower quality. I will not mention the name of the manufacturer. But, generally, what we do is we buy them. They are quite monodisperse, but we will apply electron microscopy ourselves to validate the size. Yeah, these hops, yeah, they were published recently in a separate paper. Just a nice add-on to validate that our Lie theory is also correct in the low refractive index range. But, so I can show this. So here are the hollow silica beads. They are hollow, meaning only the shell is of silica, and yeah, the core of these beads is filled with the medium. So that resembles how EV scatter light, and we use them to show that our Lie theory approximations are also, or calculations are also right for low refractive index particles like EVs.
Okay, Stepan wants to know if he can get access to your mathematical model source code, and has some specific questions about the apogee instrument. What's the state of me theory models and source code?
Yeah, so for the Lie theory, we use a MATLAB script. It's a script, in fact, which you can download. I have to think. What the name? Metzler, thank you. It's a code of Metzler et al. So with the code of Metzler et al, you can calculate angle-dependent light scattering. What we add to this script is integration of the collection angles of the flow set order. At the moment, we don't make this publicly available because the scripts are partly commercialized by the company Cytometry. Yeah, the questions about the collection angles of the apogee. What were the questions?
So it's asking if the difference between the A50s and the A60s are very large. I think a slide compared one being 4 degrees and the other being 40.
Yeah, right. That's this difference, right?
Yeah.
I didn't sketch it here on the slides, but the apogee does not have two lenses to collect the light scattering. It has only one lens. And with this lens, both forward scattering and side scattering are being collected. Then later on in the optical path, there is a mirror placed to differentiate, so to propagate these two different angles to different detectors. And the alignment of this mirror determines how much light is going to the forward scatter and the side scatter detector. So it's entirely different than any other system I have seen.
Okay. So another question about refractive index. Does cargo also affect refractive index?
Yeah, cargo does affect the refractive index, else there would not be a difference between EVs and lipoprotein particles. Whether the cargo of EVs causes a difference substantial of large enough to be measured with this technique, I don't know. We didn't study that.
All right. Esteban asks, does the refractive index change if we change the permeability? Presumably he's talking about permeabilizing somehow a vesicle.
Yeah. Presumably, yes. We didn't measure it, but I would say that.
Stephania asks, you're talking about your EVs and lipoproteins. What's the plasma from fasted or non-fasted? You see different amounts in fasted and non-fasted. Have you looked at that?
Yeah, we have looked at that, and I did not put those data on the slide. Yeah, the data I've shown today were partly from platelet concentrates and partly from a plasma sample. But before Frank Coumans left the lab, he did a funny experiment. One morning, we decided not to have breakfast at all and apply flow SR to measure our what we named the lipoprotein particle concentration. And then one day later, we changed the experiment. We ate as much as fatty food as we could in the morning. So we took a milkshake, really British breakfast with some bacon, and yeah, we found a 50-fold difference in the lipoprotein concentration. Then some of the particles larger than 1.42. Minor concentration difference in labeled movements. Slight concentration difference in the EV, so in the refractive index particles, the refractive index below 1%. So apparently, some chylomicrons end up there, and that you can also see in our results.
Okay. So looking forward, right now, you being able to use this, very elegant technique, right? Because it avoids a lot of the mathematical complexity that might slow things down. What's limiting improvement here is, will improvements in forward and side scatter sensitivity extend the range over which this is useful? Is there, a fundamental limit you see?
Yeah, that's a good question. What's the limit? This technique. So there are a few limits of this technique. Yeah, the limit is the applicable size range, and right now this is for our flow cytometer between 200 and 600 nanometers. This is partly a hardware limitation, and it's partly a limitation of the whole flow SR approach. You can imagine below 100 nanometers, this slope here becomes very flat. You can see here it becomes flat, and it becomes flat because particles do not scatter light angle dependently anymore. And you get isotropic scattering here in what we call the radial regime. So below 100 nanometer, we can mainly say the particle is below 100 nanometer. And that's probably it. Although I have no data on that because we cannot measure it. So we cannot measure particles below 200 nanometer, at least not biological particles, mainly because we cannot detect them with our forward scatter detector. With side scattering, we can. With side scattering, we can go down to about 150 nanometers at the moment, but with forward scattering, not smaller than 200 nanometers. So I would expect if we reach higher sensitivity, and especially for the forward scattering detector, the applications of flow SR would be wider than they are now. Yeah, the other limitation is here on the higher edge, at 600 nanometers. Yes, these curves start to deviate, and this is a limitation which can be solved just by changing the illumination wavelength of the laser. So here it was 400 nanometers, and then these curves start to deviate from 600 nanometers and larger. But if you would use, for example, 800 nanometer light, this whole curve would shift to the right. And just by combining two lasers, one with a long wavelength and one with a shorter wavelength, you can increase the application range of flow SR.
Okay, a couple more questions that. So Wendy asks, it has been suggested to use a neutral density filter on the SSC to quote unquote see smaller particles. Does the apogee have this, and does this even make sense? The next question from Robert is, similar vein. If you could make measurements with 3 different angles, presumably light scatter detectors, would this improve things?
So I don't know. I'm not aware of the neutral density filter trick. Yeah, it seems to me if you place a neutral density filter in front of a scatter detector, you're attenuating both the signal and the noise. So generally, it's not something I think which is really handy to do. But I may be wrong, so please send me the paper. I can check what I think. Yeah, the question about adding an extra scatter detector, I think it's very valid. Actually, there was a paper out there in 2012 of the group of Maltsev, and the paper was by Konokova et al. And they did angle-dependent light scattering measurements of single particles in a flow cytometer. So instead of 2 angles, which you see here, they measured, I think, several tens of angles. So not 3 or 4, but with really many. And so then you can resolve the whole angle-dependent light scattering of spherical particles, which I showed here on this slide. And if you can resolve this, you can inversely solve the light scattering theory and predict what was the refractive index and the size of this particle. And if you cannot solve it, it most likely means that the particle was not spherical. Yeah, that has, of course, huge advantages, analytical-wise. It has one practical disadvantage. They needed much longer integration times, so the flow cytometer was much slower than a normal flow cytometer.
Okay. Thanks again, Edwin. I think we'll wrap up there as we've managed to get through quite a lot of the questions. So we'll resume in 2 weeks' time, and this talk will be available offline within the next couple of days. So thank you, everyone that's been joining.
Thank you. The chat will be ongoing, and hopefully Edwin and others will have opportunity to continue the discussion there. Thanks, everyone, for coming. See you in a couple of weeks.